Hi everyone. This video will show you the actual experimental steps and data collected for the pH and buffer lab. Your instructor may ask you questions related to the procedure or data shown in the video. As you're going through the videos, it's useful to have the lab procedure and lab report with you so you can write down all the relevant observations. Although there are 5 parts to this lab, we will only do the first 4. Your instructor might provide you data to complete the calculations for part 5. The video will show you experiments and observations for parts A through D. Part A is about using acid-base indicators to determine the possible pH range of an unknown solution. The 5 indicators we will use are shown here, and their pH ranges are shown in Table 1 in the lab procedure. We start with a solution of 0.1 molar HCl and place 1 milliliter each in 5 test tubes. To each test tube, we added 1 drop of the indicator. This is the result for HCl, with the abbreviated names of the indicators taped to each test tube and a picture of the order of the indicators shown on the top right. We proceeded to perform the indicator test on 3 other solutions: 0.1 molar sodium dihydrogen phosphate, 0.1 molar acetic acid, and 0.1 molar zinc sulfate. Record all these results in your lab report so you can determine the pH range of each of the solutions. In Part B, our goal is to use the pH meter to measure the pH of 4 solutions: 0.1 molar sodium bisulfate, 0.1 molar sodium acetate, 0.1 molar sodium carbonate, and 0.1 molar sodium chloride. We pour about 30 milliliters each of the solutions into 4 beakers. We placed a pH electrode connected to a pH meter to measure the pH of each solution. Record the pH values you observe as follows for 0.1 molar sodium bisulfate, 0.1 molar sodium acetate, 0.1 molar sodium carbonate, and 0.1 molar sodium chloride. In Part C, we used the pH meter to measure 2 solutions of acetic acid, at 0.01 molar and at 1 molar. You should use these values to determine the acid ionization constant, or Ka, of acetic acid. In Part D, we are making a buffer using an unknown acid and its conjugate base to help us determine the Ka of the unknown acid. Although not shown here, we started by weighing about 2 grams of the solid acid and dissolving it in deionized water to 100 milliliters. The acid solution is then split into 2 50 milliliter aliquots placed in 2 separate beakers as shown here. One of the beakers is labeled HA for the acid and the other is labeled A-. The acid in the beaker labeled A- will be converted to its conjugate base by titration with 0.1 molar sodium hydroxide. To start the titration, we added 3 drops of the phenolphthalein indicator. We then titrate the acid with NaOH until we reach the equivalence point as shown by the light pink color of the solution. We then add water to the original beaker, labeled HA, to the same volume as the beaker containing the pink A- solution. As you can see, both beakers contain solutions to the same volume. Lastly, we made a buffer by mixing 25 milliliters of HA with 25 milliliters of A-. This solution is called the 50-50 buffer. The pH of the 50-50 buffer is measured in a pH meter. You can record this value in your lab report as highlighted here and use the pH to help you determine the Ka of the unknown acid.